Today we present an article from Nature Microbiology titled Endogenous Murine Microbiota. Member Faecalibaculum rodentium and its human homologue protect from intestinal tumor growth. This study reveals that an endogenous strain of the mouse microbiota, Faecalibaculum rodentium isolate PB1 and its human counterpart Holdermanella biformis, both belonging to the Erysipeloidraceae family, are lost during the early phases of tumorigenesis. These microorganisms block tumor cell proliferation by reducing the activation of nuclear factor of activated T cells, cytoplasmic 3N, FATC3 and calcineurin. First, by tracking the microbial composition of colon cancer model mice at different stages, the research team identified a strain named F. rodentium. This taxonomic unit did not expand in A-twickled APC mine divided by mice, coinciding with the onset of tumor development. Subsequently, a strain belonging to this taxonomic unit was isolated and fully sequenced from WT mice, revealing it to be the sole representative of F. rodentium within the WT mouse population. The previously uncharacterized FPB1 isolate was associated with mucus in both the small and large intestines. It was drastically reduced in the mucus of APC min plus divided by plus mice. Thus, a F. rodentium strain was identified that is typically abundant in WT mice, but severely underrepresented in APC min plus divided by plus mice during early tumorigenesis. They next tested whether FPB1 was capable of colonizing APC min divided by plus mice. To eliminate competing microorganisms, they treated both WT and APC min divided by plus mice with antibiotics and administered FPB1 via gavage. FPB1 abundance was lower in the ileal mucus of APC min divided by plus mice and WT mice at 48 hours, suggesting that the APC min divided by plus gut is unfavorable for FPB1 colonization. When validating F's effect on tumor growth, it was found that only at 8-12 weeks did F exhibit a significant impact on tumor growth. The research team subsequently evaluated the impact of this bacterial population on immune cells such as CD4+, neutrophils, and monocytes, but did not identify any significant correlations. They then analyzed alterations in microbial composition and metabolites, and results suggest that FPB1 affects tumor cell proliferation either independently or in cooperation with other bacteria, likely through the release of short-chain fatty acids Fs. Butyrate exhibits histone deacetylase HDAC inhibitory activity, which can influence cell proliferation. Unlike normal cells, colorectal cancer CRC cells do not utilize butyrate, leading to its accumulation and exertion of HDAC inhibitory effects. Studies indicate that 1-2 millimeters butyrate, along with higher concentrations of acetate and propionate, inhibit proliferation in multiple mouse intestinal tumor cell lines, with short-chain fatty acids Fs exhibiting cumulative inhibitory effects. Further investigations revealed that under strict anaerobic conditions, strain FPB1 efficiently produces butyrate and lactate, with butyrate concentrations approaching those effective for proliferation inhibition, while acetate and propionate levels remain extremely low. FPB1 culture supernatant significantly suppress tumor cell proliferation without affecting cell viability, indicating that released metabolites exert antiproliferative effects. Mechanistic studies revealed that FPB1 sup or SCFA combinations enhance histone H3 acetylation H3K27AC, confirming its HDAC inhibitory activity, and downregulate calcineurin PP2B and NFATC3 activation. After evaporative removal of Fs, sub's ability to induce H3 acetylation and downregulate NFATC3 was significantly weakened, indicating this effect is primarily mediated by Fs like butyrate, with minimal lactate involvement. In summary, FPB1 inhibits tumor cell proliferation by producing Fs, especially butyrate, to suppress DAX and block the calcineurin divided by NFATC3 pathway. Next, we assessed whether butyrate alone could mediate the antiproliferative response. 1 mm sodium butyrate matching the concentration found in F. Hmone sup was administered to antibiotic-treated APC mine divided by plus mice to avoid potential consumption by the local microbiota. Butyrate exhibited antiproliferative activity highly similar to that of F. Hb1 sup, indicating butyrate as the primary effector of F. Hb1 activity. Collectively, these data demonstrate that F-Hb1 metabolites, particularly butyrate, due to their Hitachi activity, control NF-ATC3 activation, thereby blocking tumor cell proliferation in APC mine divided by plus mice independently of the microbiota. Similar antiproliferative activity was observed in inflammation-driven CRC models. They then evaluated whether F-Hmone's activity is shared by other SCFA-producing bacteria. Lactococcus lactis was selected because it secretes butyrate and some lactate in culture, similar to F-Hb1, and has been proposed to exhibit antiproliferative activities in vitro via an unknown mechanism. They administered L. lactis following a schedule similar to that of F-Hb1, but could not detect any anti-tumor effect. This is likely due to L. lactis' inability to survive or colonize the mouse intestinius. They could not detect L. lactis even in WT mice independently of the microbiota. They thus analyzed the antiproliferative activity of the sup from L. lactis in vivo, antibiotic-treated APC mine divided by plus mice. They found that the L. lactis sup reduced the size of tumor lesions, although not as effectively as the F-Hb1 sup. This difference cannot be due to a reduction in lactate production, as lactate did not impact tumor cell proliferation in vitro. The antiproliferative activity of F-Hb1 is therefore not unique to this bacterium, and may be common to other bacteria that produce this, provided that they are able to colonize and divide by or survive in the intestine long enough to produce butyrate. That's all. Thanks for your listening.